Warm greetings to all the delegates and faculty members. It's nice to be a part of this academic feast again. Let me start sharing my screen with you. I am Dr. Krishna HM, and the topic for today's lecture is Applied Physiology of Carbon Dioxide Transport. The lecture objectives would be to find out how the carbon dioxide transport occurs within the body, to understand the carbon dioxide dissociation curve, the differences between venous and arterial blood with respect to carbon dioxide transport, and the influence of carbon dioxide on acid-base homeostasis. As we all know, carbon dioxide is produced in almost all the cells of the body as end product of metabolism. And in the tissues, as the carbon dioxide level concentration builds up, it just moves out of the tissues into the capillaries which are perfusing the tissues along the concentration gradient. So about 200 ml every minute of carbon dioxide is produced in the body in the resting state. And this moves out of the tissues into the capillaries along its concentration gradient. This is the dissolved form of carbon dioxide. Though in this form, it might not be contributing significantly to the amount of carbon dioxide that is being transported from tissues to the lungs, we have to remember the importance of this dissolved form of carbon dioxide, because this is the only form in which carbon dioxide can move across the cell membranes. Whether it is from the tissues, cells to the blood, or from the blood across the alveolo-capillary membrane into the alveoli. There's one added advantage to carbon dioxide here, in that it is 20 times more soluble than oxygen in the blood. Once carbon dioxide enters into the blood, it can remain in the blood, in the plasma, in the dissolved state itself, and then move along the circulation to the pulmonary circulation, and then across the alveolo-capillary membrane and get exhaled from the lungs. But the fraction of carbon dioxide which is transported in this form, that is the dissolved carbon dioxide, is quite less. It's only about 5%. The other way in which the carbon dioxide is transported in the blood is in combination with the plasma proteins. There are several plasma proteins. We know albumins are there, globulins are there. And these plasma proteins have amino acids which have an amine end in their chain. So to this amine end, the carbon dioxide combines to form carbamino compounds. That's another way of transport of carbon dioxide. But the majority of the carbon dioxide which enters the blood from the tissues simply move into the red blood cells. So about 75% of carbon dioxide is carried in the erythrocytes. Within the erythrocytes, again, carbon dioxide can remain in three forms. One is again in the dissolved form. That's how the carbon dioxide enters from the blood into the red blood cell. But again, it remains in the dissolved state only in a small proportion. It can combine with the hemoglobin, the globin moiety of the hemoglobin, which is again a protein, to form a carbamino compound. But the third form is the majority, that is majority of carbon dioxide undergoes a hydration reaction within the red blood cell. It combines with water to form carbonic acid, which dissociates into hydrogen ion and bicarbonate. Now we don't see this reaction happening predominantly in the blood or plasma, because within the red blood cell there is a unique enzyme which catalyzes this reaction, that is the carbonic anhydrase enzyme. Because of the carbonic anhydrase enzyme, the carbon dioxide which enters the red blood cell within a fraction of a second dissociates into hydrogen ion and bicarbonate ion. We must pay attention here that in this equation, the arrow marks are bidirectional. That is, over a period of time, if the hydrogen and bicarbonate ion concentration keeps increasing in the red blood cell, then that will trigger the reaction to happen in the opposite direction. It will again combine to form carbon dioxide and water. This doesn't happen in reality because the hemoglobin readily combines with the hydrogen ion. If you look at the tissue level, the hemoglobin has come to the tissues. It was loaded with oxygen. Now it has given away the oxygen to the tissues, so it's in a reduced state, and the reduced state of hemoglobin has a higher affinity for the hydrogen ion. It readily combines with the hydrogen ion. So there is no buildup of hydrogen ion within the red blood cell. Now the bicarbonate within the red blood cell is sensed by an ion transporter which is present on the membrane of red blood cell and is actively transported out of the red blood cell into the plasma in exchange with a chloride ion to maintain the electric and osmotic neutrality within the red blood cell. So bicarbonate is pumped out, and in exchange chloride is pumped in by this ion transport. This is called as chloride shift, or popularly what is called as Hamburger phenomenon or reaction. So there is no accumulation of hydrogen ions or bicarbonate ions within the red blood cell, and hence almost any amount of carbon dioxide which enters the red blood cell can be hydrated to yield hydrogen and bicarbonate ion, and it is in this form that carbon dioxide is predominantly transported in the blood to the pulmonary circulation for its final destination into the alveolus. So far we saw that carbon dioxide is transported in three forms, three methods. One is the dissolved carbon dioxide. Percentage-wise, the contribution is quite less, but this is important because this is the only form by which it can move across the cellular membranes. The second one we saw was the majority of the transport of carbon dioxide which occurs within the red blood cell following hydration of carbon dioxide yielding carbonic acid catalyzed by carbonic anhydrase enzyme and then resulting in hydrogen ion and bicarbonate. And the third form that we spoke, which happened even in the plasma, is combination of the carbon dioxide with the proteins. Within the red blood cell, it's going to be the hemoglobin. So there are histidine moieties within the globin chains, and this is readily available for combination with the carbon dioxide. So at the amine end of the amino acid, the carbon dioxide readily combines to form the carbamino compound. Though we spoke of the percentages in which carbon dioxide is transported in the red blood cell in this form, if you actually see the composition of carbon dioxide which is exhaled, the contribution from these three will change. In fact, there is much more contribution from the carbon dioxide which is transported in combination with the proteins. The blood which reaches the pulmonary circulation, we know that gas exchange is occurring there. Oxygen moves into the pulmonary circulation and carbon dioxide moves out of the pulmonary capillaries into the alveoli. So the oxygen tension in the blood, the PCO2, reduces there. Remember, this dissolved carbon dioxide is always in equilibrium with the other forms of carbon dioxide, be it combination with the proteins or be it combination with the protein in the form of hemoglobin or in the form of hydrogen ion and bicarbonate. So there's always an equilibrium. So once the dissolved carbon dioxide moves into the alveolus, the concentration reduces, and hence whatever reactions we saw in the red blood cell at the tissue level, the reverse starts happening within the red blood cell. So what happens there? There is abundance of oxygen coming from the alveoli, and hence hemoglobin readily gives up the hydrogen ion and now takes up the oxygen along with that. So the hydrogen facilitates the uptake of oxygen by the hemoglobin, and once one molecule combines, that will further facilitate the uptake of other three molecules by the hemoglobin. Once the concentration of hydrogen ion in the red blood cell increases, this is sensed by the ion transporter molecule. It actively pumps in bicarbonate into the red blood cell from the plasma in exchange to the chloride which was present in the red blood cell. The hydrogen ion and the bicarbonate now readily combine to form carbonic acid, which dissociates into carbon dioxide and water. Carbon dioxide moves out of the red blood cell into the plasma and then along the concentration gradient into the alveoli. So this is what happens at the level of the pulmonary circulation, and thus carbon dioxide is eliminated from the body. In relation to carbon dioxide transport, we speak of an interesting phenomenon, an interesting effect called as Haldane effect. It describes the increased ability of blood to carry carbon dioxide as hemoglobin offloads its oxygen. So when the hemoglobin comes at the tissues, it gives away the oxygen, and this increases the ability of the blood to further carry carbon dioxide from the tissues. There are two reasons behind the Haldane effect. The deoxygenated hemoglobin is 3.5 times more effective in forming carbamino compound with carbon dioxide. So, because it is a very good proton acceptor, the deoxygenated hemoglobin readily combines with hydrogen ion, buffers it very easily, better than what an oxygenated hemoglobin would have done. So the first reason contributes to about 70% of the Haldane effect, whereas the second reason, that is the enhanced binding of hydrogen ion, contributes to about 30% of the Haldane effect. So in essence, the ability of hemoglobin to bind carbon dioxide to form carbamino compound is enhanced, and the ability of deoxygenated hemoglobin to bind hydrogen ion is also enhanced. So dash concentration determines hemoglobin's affinity for dash. If it is Haldane effect, it is the oxygen concentration which determines hemoglobin's affinity for carbon dioxide. Synergistic with this, remember hemoglobin is also carrying oxygen. We have the Bohr effect. Dash concentrations determine hemoglobin's affinity for dash. So it's the carbon dioxide and hydrogen ion concentration which determine the hemoglobin's affinity for oxygen. So it is sort of a reciprocal and happening in harmony, in synchrony, both at the tissue level and at the pulmonary level, one enhancing the other. So if we were to see at the tissue level, the binding of carbon dioxide is enhanced by giving up of the oxygen. Now this actually happens at the molecular level by virtue of the configurational change of the hemoglobin molecule, what is in a very complex way described as heterotropic allosteric ligand interaction. It means nothing but the positional change of the polypeptide chains will give it a property of enhanced binding or reduced binding. Similarly, at the lung level, the oxygen uptake is facilitated, carbon dioxide and hydrogen ions are released. If we pay attention to carbon dioxide dissociation curve, no, I'm not sure whether it should be called a dissociation curve because it's more of a relationship curve between the partial pressure of carbon dioxide and carbon dioxide concentration in the blood. On the x-axis, we plot the PCO2. On the y-axis, we plot the carbon dioxide concentration. Remember, in your oxygen hemoglobin dissociation curve, you had the oxygen saturation of hemoglobin on the y-axis. As we have already mentioned during the transport of carbon dioxide in the blood, that if carbon dioxide were to be transported only in its dissolved form, then the body wouldn't have enough capacity to eliminate all the carbon dioxide that is synthesized in the body. As this graph depicts, even at the physiological pH of 35 to 45 millimeters of mercury, what would be carried in the blood would be only about, say, 5 to 6 ml per deciliter of carbon dioxide, which is grossly insufficient. But fortunately, we have the other mechanisms of formation of carbamino compound and the contribution of red blood cell and the hydration reaction, which will ensure that what needs to be eliminated is eliminated in the physiological range of PCO2. If you see the black box in this graph, that's where the human beings are actually operating. So the pH, the physiological range of PCO2 of 35 to 45 millimeters of mercury. And in that, you will notice that the carbon dioxide dissociation curve is linear. Now, whatever is to the left or right of this graph actually is not happening in a normal human being. It's only extrapolations that are obtained under experimental conditions. You see that contrary to the oxygen hemoglobin dissociation curve, this curve is more or less linear. You had a sigmoid shaped in oxygen hemoglobin dissociation curve, whereas here it is linear in the sense that if the PCO2 keeps increasing, the oxygen concentration in the blood also can keep increasing. There is no plateau effect. And this is because of the huge, immense capacity of the hydration reaction that is happening within the red blood cell. You also notice that there are two separate lines or graphs for deoxygenated blood and the oxygenated blood. The reason for these two curves that we are getting is purely because of the Haldane effect, because of which the carbon dioxide carrying the capacity of the venous blood, deoxygenated blood, is enhanced. So at a similar partial pressure, the venous blood would have a higher concentration of carbon dioxide as compared to oxygenated blood. Now, what's the difference between venous and arterial blood with respect to carbon dioxide transport? As we have already mentioned, the content of carbon dioxide in the venous blood is higher than that in the arterial blood. It's about 52 ml per deciliter in the venous blood. Very obvious reason, tissues are producing carbon dioxide and that moves into the blood and that is efficiently taken up and transported by the venous blood. And this property is given to the venous blood because of an enhanced capacity to hold the carbon dioxide in the venous blood, which is deoxygenated by virtue of the Haldane effect. Because of the carbon dioxide hydration reaction within the red blood cell, you need to notice that for every molecule of carbon dioxide, there is one molecule of bicarbonate or chloride, which is osmotically active, which is present in the red blood cell. This osmotically active particle will draw in some amount of water into the red blood cell in the venous side and cause an enlargement of the red blood cell, which results in the increase in the hematocrit of venous blood by about 3% as compared to the arterial blood. Now, the hydration reaction of carbon dioxide at tissues result in large hydrogen load, but this is not reflected in the changes in the pH. Simple reason is the reverse of the hydration reaction is happening at the lungs and carbon dioxide is continuously eliminated. So there is no net essential addition of hydrogen ion within the circulation. And the other reason is that whatever hydrogen ion is formed by the hydration reaction, it doesn't remain as a free hydrogen ion. It is immediately very nicely buffered by the deoxygenated hemoglobin. It just grabs and holds and binds that hydrogen ion, so it doesn't cause any change in the acid-base balance. So, though we see that the venous blood is slightly more acidic than the arterial blood, be it arterial blood or venous blood, the pH would still remain in the physiologic pH range of 7.35 to 7.45. It's only in the pathologic condition when the carbon dioxide is not getting eliminated or there is increased synthesis or whatever is causing hypercapnia that these physiological mechanisms are overwhelmed, the buffering capacity is overwhelmed, that gets reflected in the form of respiratory acidosis. On the contrary, if the carbon dioxide levels are lower, we would have respiratory alkalosis. This carbon dioxide in general across the tissues in the body would cause vasodilation and the effects secondary to vasodilation. So if you were just to take an example of the cerebral circulation, it causes cerebral vasodilation, which would result in raised intracranial pressure. So similarly, the effects at various tissue levels can be deciphered by this general principle that it is usually a vasodilator, except probably in pulmonary circulation, where it can cause pulmonary vasoconstriction. Carbon dioxide also, in general, stimulates the sympathetic nervous system. And you can see the effects of stimulated sympathetic nervous system like tachycardia, hypertension. To summarize, we saw the three modes of carbon dioxide transport: the dissolved form, the carbamino compound formation, and the hydration reaction resulting in hydrogen and bicarbonate ion, and the efficiency with which this transport occurs. The Haldane effect, which enhances the capacity to carry the carbon dioxide, and this is influenced by the oxygen levels. We saw the carbon dioxide dissociation curve and its properties, the influence of carbon dioxide on the acid-base homeostasis. I thank you very much